Welcome to the third video lecture of muscle tissue. In this video lecture, we're going to cover some real basics of muscle physiology, including an introduction to the sliding filament model or mechanism of contraction. We're going to talk about exactly what causes muscle cell excitation, or you could call that the events at the neuromuscular junction. We're going to examine what will connect that activation or excitement at the neuromuscular junction to the contraction cycle. So we're going to be examining something called excitation-contraction coupling. And then finally, we're going to look at the events of contraction. Blow by blow, what happens as your filaments slide past each other. But let's begin with a basic introduction to the sliding filament model of how muscle cells do contract. So the sliding filament model is our best current explanation of how a muscle cell can generate force. Now, just because a muscle cell generates force doesn't mean that the fiber physically shortens. Sometimes you may pick something up and you may simply hold it in place. You may do what's sometimes called an isometric exercise. That means that you are generating force, but the length of the muscle isn't changing, which means that the length of the individual muscle cells is not changing, but still you're generating force. Now, you do shorten the muscle or shorten the muscle cell when the force or the tension that's generated by all the cross bridges between the thick filaments and the thin filaments, when all of that force exceeds the forces that oppose shortening. Or you could think of it as when you generate enough force with your muscle that it overcomes the force of whatever weight you're trying to move. And then the muscles will lengthen or shorten and move. So here's what we mean when we talk about the sliding filament model of contraction. Really what we're going to be looking at is overlap between the thin and the thick filaments. So if you look up at step 1 of this, you'll notice that the thin filaments in blue and the thick filaments in orange have only a little amount of overlap. And if you look at the transmission electron microscope picture above that, the two dark lines on either side are the Z discs, and then the dark area in the center is the A band where you see the thick filaments. The lighter area is the I band, and you can see that there's a lot of I band. And you can also see an area of the H zone in the center where there's a little bit of lightening in the center of the I band. Where there's a little bit of lightening in the center of the A band. Now look down at step 2. This is a fully contracted sarcomere. One thing you may notice is this is the same shot, but now the Z discs are a little bit closer together. So the sarcomere has shortened. How did this happen? Well, if you look down at the drawing, you'll see that there is an increase in the amount of overlap between the thin filament and the thick filament. In fact, if you look at the blue thin filament, it's actually at the M line, at the very center of the sarcomere. Now let's look up at our electron microscope picture, and we can see that the Z discs are closer together. There's less total area of the I band. So that's the area where you see only thin filament. If you look at the thick filament, it's about the same size. But look in the center of the thick filament. You don't see a distinct H zone, and that's because the overlap between thin and thick has gone all the way across the A band. So in the relaxed state, the thin and the thick filaments only overlap towards the ends of that A band. There's only a little bit of overlap between thin and thick filaments. In the sliding filament model, the thin filaments will slide past the thick filaments, so that means that the actin and the myosin overlap even more. This overlap, or this movement, occurs when the heads of the myosin bind to the actin. This forms a cross bridge, and the myosin heads literally push the actin in the thin filaments towards the center of the sarcomere, towards that M line. It draws them inward. So once again, here we see our fully relaxed sarcomere. You can see that there's a little bit of overlap between the thin and the thick filament. All of those little knobby bits of the thick filaments in orange in our drawing would be able to reach out and contact the actin, but in this relaxed sarcomere, there's really no cross bridges. There's very little amount of overlap. All right, but we see a large area of the I band where you have the light area, because that's where you find only thin filaments. Then the myosin heads will bind to the actin. Sliding begins, those cross bridges form, so they form and break and form and break, and it makes this little ratcheting action that will inch or push slowly the thin filament towards the center of the sarcomere. When that happens, the entire muscle fiber will shorten. The Z discs come closer together. They get closer to the M line. As each individual sarcomere gets shorter and shorter, that means that the entire myofibril gets shorter, because the myofibril is attached to the sarcolemma, the muscle cell membrane, by that protein called dystrophin. You're also pulling on the entire muscle cell, the entire myofiber. So the entire muscle fiber gets shorter. I bands get smaller, Z discs come closer together, H zone disappears because the H zone is where you don't have overlap between thin and thick filaments. And the A band will move a little bit closer, but the length of the A band will be exactly the same. So here, once again, we can see that fully contracted sarcomere. The Z disc is closer to the M line. There's a maximal amount of overlap between the thin and the thick filament. The thin filaments are actually overlapping, beginning to overlap with each other at the M line. So there's no area in the center where you only have thick filament. There's no H zone. Again, one more picture so that you can look at and see the difference between the relaxed sarcomere at the top and the contracted sarcomere at the bottom. If you want to look at this in a little more detail in your textbook, it's figure 9.6, the sliding filament model of contraction. All right, so that's the sliding filament mechanism. That is physically how a muscle fiber will contract, but how do we get there? How do we cause this muscle contraction to occur? There are two things you need to happen to trigger that muscle contraction. The first is activation. This is excitation. Remember, that was one of the characteristics of muscle fibers. It is excitable. It is irritable. You can activate it. And it's activated by the nervous system. This is skeletal muscle. Skeletal muscle must be activated by the nervous system. This activation will occur in an area called the neuromuscular junction, and the name makes sense. It's the connection point between the neuron and the muscle cell. During this activation, there will be an action potential generated in the sarcolemma. Now, an action potential is a change in the electrical charge across the membrane. Normal skeletal muscle cells are polarized. That means there's a difference in the number of positive and negative charges across the membrane. But when it becomes activated, that polarization changes. So we change the electrical activity across the muscle cell membrane, and then that spreads across the entire sarcolemma. So when that change in electrical activity of the membrane spreads, that's known as an action potential. So that's going to stimulate the muscle cell, but how do we connect that to actual contraction? And that's the steps associated with excitation-contraction coupling. The action potential spreads across the sarcolemma, and it goes all the way down into the T tubules. This change in electrical charge across the membrane and down in the T tubules will cause the sarcoplasmic reticulum to release calcium. So the calcium levels inside the cell, intracellular calcium levels, rise fairly quickly but briefly. So let's look, this is a figure out of your textbook, figure 9.7, and it just details the different phases of excitation, events at the neuromuscular junction, that's phase 1, and then phase 2, excitation, contraction, coupling. All right, so when we look up at phase 1, we're going to go through all of these steps individually as we go through the next several slides. And then phase 2, we're also going to be going through these steps individually. But remember, you can refer back to figure 9.7 in your book to see this nice outline view, or really a flowchart type view. All right, so let's start with events at the neuromuscular junction. So we know that skeletal muscles must be stimulated by the nervous system. The types of cells in the nervous system that physically stimulate skeletal muscle are called somatic motor neurons. Motor neurons are neurons that come out of the central nervous system carrying motor commands, telling the body what to do. The term somatic means that it's going to be controlling the movement of the body. That's skeletal muscle. So skeletal muscles are stimulated by somatic motor neurons. Axons from those somatic motor neurons travel from the central nervous system, either the brain or the spinal cord, and will go out via nerves to the skeletal muscles. Each of those may branch several times as it gets into the muscle, but eventually the endings of those neurons, the axons, will form a junction with a single muscle fiber, a neuromuscular junction. Usually just one, but sometimes there are multiple. If it's a very long, very large muscle fiber, there may be several neuromuscular junctions associated with that muscle fiber. If it's a very small one, there may only be one. So here's just a general picture of this neuromuscular junction. You see the axon from a neuron coming in. There's a little swelled area at the end of the axon. The axon is in yellow in this diagram. And then it contacts the muscle cell, and it's going to cause a change to occur inside the muscle cell. So let's look a little bit more at this neuromuscular junction. Usually if there's just one, it's going to be about midway along the length of the muscle fiber. If there's two or more, they'll be usually spread evenly out across the entire muscle fiber. The neuromuscular junction consists of at the end of the axon, so it's called the axon terminal, and the muscle fiber. But those two structures are separated by a little space filled with some gel known as the synaptic cleft. So it's a synapse. A synapse is a connection between a neuron and some other cell. So this is a cleft or opening at that synapse. The axon terminal will contain synaptic vesicles. Now a vesicle is just a membrane sac. This membrane sac inside the axon contains a chemical known as neurotransmitter. In the case of the neuromuscular junction, for skeletal muscle, the neurotransmitter will always be acetylcholine, which is abbreviated ACH. This is going to eventually be released from the end of the axon terminal. It's going to go across the synaptic cleft, and then it's going to contact the sarcolemma. And if we look at the sarcolemma, and we will, there's going to be little folds of the sarcolemma containing receptors for acetylcholine. So the neuromuscular junction will include the axon terminals, the synaptic cleft, the junctional folds, which contain the acetylcholine receptors. All right, so let's move through and see what happens when a nervous impulse comes down the axon of a somatic motor neuron. All right, so first we're going to start with step one. The action potential arrives at the axon terminal. So it's coming, an action potential is a nervous impulse. It comes down the somatic motor neuron axon until it reaches the end, the axon terminal. When it reaches that axon terminal, it will open up voltage-gated calcium channels. Now, back in chapter 3, we went over different types of gated channels. So let's just review them real quickly. Gated channels are channels that will open or close when they are stimulated to do that. Now, there might also be some leakage channels that are just always open, but a gated channel is stimulated to open. There are many ways to stimulate a gated channel. You could have a ligand or chemically gated channel. That means you have to add a chemical to this ion channel and then it will open up. A channel could be mechanically gated. That means if you change the shape of the membrane, that will stimulate the channel to open up, like pressure on your skin will cause the opening of some mechanically gated channels. And then here we see an example of a voltage-gated channel. And this is a voltage-gated calcium channel. So when we have this change in electrical potential across the membrane, and that's an action potential, it opens up voltage-gated calcium channels. Calcium will enter the axon terminal, moving down its concentration gradient and being pulled in a little bit by the electrical current as well. So when calcium enters the axon terminal, it causes the synaptic vesicles to fuse with the outside, and that is going to release it will release acetylcholine by exocytosis. So the calcium is the trigger for the release of acetylcholine by exocytosis. The acetylcholine will diffuse from the axon terminal across the synaptic cleft and eventually will bind to receptors that are found on the sarcolemma, on those junctional folds of the sarcolemma. And those folds really just increase the surface area where you could find those acetylcholine receptors. So acetylcholine will bind to that receptor. As it binds to that receptor, it will open up. So this is actually a form of a chemical or ligand gated channel. It will cause this ion channel to open up and it allows positive ions to pass through it. Now, normally the inside of the cell has very little sodium in it and it's relatively negatively charged. So because it has very little sodium and it is negatively charged, literally the sodium rushes into the cell. Positive charges get attracted to negative charges. So remember, the inside of the cell is going to be negatively charged. The outside of the cell is going to be positively charged. And so these will rush into the cell. All right. Now, potassium will have a tendency to leave, but far more sodium is going to come in than potassium will leave. All right. But this is going to be enough that we begin to change the electrical charge across the membrane. The inside starts to become much more positive compared to the outside. All right. So when acetylcholine binds, it opens up those channels, the ions rush across, we rapidly get a change in the electricity of the membrane, but we need to be able to control this. So very quickly, the acetylcholine is broken down by an enzyme that's also found at the synaptic cleft, and this enzyme is known as acetylcholinesterase. Remember, enzymes are named after what they break down. So acetylcholinesterase will break down acetylcholine. and then the broken parts will diffuse away from the junction. But because we no longer have acetylcholine, that ion channel closes and the ions can't pass across anymore. So here we just see all of this from beginning to end. We get our action potential from our somatic motor neuron. Calcium rushes into the axon terminal. We have exocytosis of acetylcholine from the synaptic vesicles. Acetylcholine diffuses across the synapse, attaches to those receptors, those ligand-gated channels, opens up, allows sodium and potassium to rush across, but a lot of positive charges enter the cell, and then the effect is stopped by acetylcholinesterase breaking down acetylcholine. So it's deactivated, breaks the acetylcholine down into acetic acid and choline. So that means that the muscle fiber will contract very briefly in response to a single nervous impulse from the somatic motor neuron. To get sustained muscle contraction, you've got to have repeated stimulation. You've got to have more than one nervous impulse going down that somatic motor neuron. And we'll see what happens when you do have those repeated stimuli. All right, so let's talk a little bit more about this thing we call an action potential. The resting sarcolemma cell membrane is polarized. That means that there are lots of negatively charged particles inside the cell compared to the outside. Positive, or anything that's charged, has a hard time making its way across the membrane. The membrane will not allow positive or negative charged ions to pass across it. So we separate out this electricity, and we call this the voltage. It's just like a battery. Batteries that you buy have certain voltages associated with them. The two poles of the batteries are positive and negative, and they are separated from each other. It's not until you open up or you connect the poles of the battery that you actually get any work from the battery. And that's what happens with an action potential. We open up spaces between the inside of the cell and the outside of the cell by those channels. And ions now can rush into the cell. Those positively charged sodiums can rush into the cell. So we connect essentially the two sides of the battery, and we get work associated with this. And we call this depolarization. So as those positive charges come in, we started off polarized, and now we unpolarize the membrane. That's the stimulation, that depolarization. And then we inactivate it by the acetylcholinesterase, and the cell will repolarize. Now that depolarization will propagate, and the term propagate just means to spread. So that's our action potential. The depolarization spreading over the membrane and down into the deep portions of the cell through those T tubules. All right, so that's excitation. Somehow we've got to connect this with the contraction cycle, but really right now we've only talked about things occurring at the membrane. We've got to somehow cause a change to occur inside the cell. So the events that transmit the action potential, AP stands for action potential, along the sarcolemma will eventually lead to the sliding filament mechanism. That action potential is brief. It actually ends before contraction starts, but what it does cause is a rise in the intracellular calcium levels. And the intracellular calcium is what triggers contraction. It leads to contraction. So this brief period of time before contraction, before you get any force generated, is known as the latent period. And we're going to come back to that when we talk about a single twitch contraction. But this latent period is the time during this excitation contraction coupling. Before you get contraction, but what will allow you to have contraction. So it's a time between the beginning of the action potential and the beginning of contraction. This is the time period of excitation-contraction coupling. So there are several events. The action potential spreads along the sarcolemma to the T tubules. All right. Voltage-sensitive proteins will stimulate in voltage-sensitive proteins on the sarcoplasmic reticulum will detect the change in voltage. Now remember, those T tubules were laying right alongside sarcoplasmic reticulum. So the sarcoplasmic reticulum would easily be able to detect the change in voltage. And that's going to stimulate the sarcoplasmic reticulum to release calcium. And calcium is the trigger for contraction. So here we see setting the stage for contraction. We had the events associated with excitation at the neuromuscular junction, and now we've got to spread this across the sarcolemma, down the T tubules, and allow the release of calcium from the sarcoplasmic reticulum. So we have the change in electricity spreads across the sarcolemma. The T tubules are really an extension of the sarcolemma. It spreads down those T tubules, and right next to those T tubules, you have sarcoplasmic reticulum. It's sensitive to the voltage. Channels will open up for calcium to be released, and calcium will begin to flood out of the sarcoplasmic reticulum. So the calcium ions are released, and those calcium ions, they're going to be the trigger for contraction cycle. So let's go back and have a look at our thin filament in blue and our thick filament in orange, and we're only looking at a small area. All right, so in this thin filament in blue, you see that the tropomyosin of the tropomyosin-troponin complex is covering those binding sites, those active sites. So that means that you can't get contraction occurring. All right, so the calcium is going to be the trigger for this contraction. When calcium is there, it will attach to the troponin, but it moves the tropomyosin, and now all of a sudden those myosin binding sites are exposed. Those active areas are exposed and ready for myosin to bind, and that's going to begin the contraction. All right, so contraction will begin when the myosin head forms a cross bridge. It attaches to the thin filament, and we're going to begin to move the thin filament. All right, so really brief summary once again. We have the events at the neuromuscular junction, excitation, causes an action potential. The action potential spreads, then we have excitation-contraction coupling, which is the action potential spreading or propagating across the sarcolemma. It moves down into the T tubules and causes voltage-sensitive channels in the sarcoplasmic reticulum to open up and release calcium. Then that's going to be the trigger for the contraction cycle. All right. So there's a lot of different channels that are involved across the membrane for initiating muscle contraction. All right. So first off, when the nerve impulse reaches the axon terminal, we had those voltage-gated calcium channels, and that allowed acetylcholine to be released into the synaptic cleft. Another channel was where the acetylcholine bound to its receptors. Those were ligand-gated channels, and they allowed positive charges, sodium and potassium, to pass through them. And that caused the change in electricity right at the neuromuscular junction. We call that the end plate. But that will open voltage-gated sodium channels on most of the sarcolemma. And that's where we get an action potential, because the voltage-gated channels will continue to open, and that will spread the action potential across the entire sarcolemma and down into the T tubules. Then we have voltage sensitive proteins that are found between the T tubules and the sarcoplasmic reticulum, and that's going to release some ion, open up some ion channels in the sarcoplasmic reticulum, releasing calcium out to the cytosol. So, role of calcium in contraction. Physiologically, what does calcium do? If the calcium levels are really low inside the cell, the tropomyosin covers the active sites on the actin, so the myosin can't attach, the muscle fiber is relaxed. But when the calcium levels go up right around those sarcomeres, the calcium binds to the troponin. Troponin changes shape a little bit. Tropomyosin moves away from the myosin binding sites. The myosin heads bind to the actin, and that triggers contraction. So the sarcomere will begin to shorten. You'll begin to get some force generated, and you can get muscle contraction. Whenever that nervous stimulus ends, when the acetylcholine is broken down by the acetylcholinesterase, then all of this activity ends. The action potential ends. The sarcoplasmic reticulum no longer releases calcium, so the sarcoplasmic reticulum, the SR, begins to pump calcium back into it. Once the intracellular calcium levels drop, then you cover the myosin binding sites again, and you get relaxation. So all of these are events that lead up to the actual sliding filament mechanism. And there is a series of events that occur to allow the sliding filament mechanism to occur. So these are events associated with physically getting contraction with that sliding filament mechanism. And it will continue as long as calcium is around and as long as you have enough ATP around. One essential ingredient of this contraction cycle or cross bridge cycle is the formation of the cross bridges, where the myosin heads that have been activated by ATP attach to the thin filament. Once they attach to the thin filament, the myosin heads are going to pivot, and that will pull or push the thin filament towards the center of the sarcomere, towards that M line. But if you want to repeat this cycle, you've got to break those cross bridges. So cross bridges have to detach, and they do this by ATP attaching to the myosin head, and that causes the cross bridge to break apart. Then the myosin head will hydrolyze the ATP. It will break the ATP into ADP, and that moves the myosin head back into a high energy state. Sometimes this is called cocking of the myosin head. So let's look at some of these events. So if that myosin head is already back in a ready position, that means it's already activated. ATP has been broken down into ADP plus an inorganic phosphate. As soon as we have calcium around, you can have contraction. So first we get cross bridge formation. The energized myosin head attaches to an actin myofilament, and we get our cross bridge. Then we have this working stroke. The ADP is released, and the myosin head will pivot just a little bit. It's still attached to the thin filament. So as it pivots just a little bit, it pushes that thin filament towards the M line. Then we add another ATP to the myosin head, and that breaks the myosin head off of the actin, off of the thin filament. When it hydrolyzes that ATP, it moves back once again to that ready position, to that cocked position, and as long as those binding sites on actin are still available, we're going to keep on getting this cross bridge formation, power stroke, and we're just going to keep on going. So this entire cycle will continue as long as you have plenty of ATP available and as long as calcium remains bound to troponin. All right, so that does it for some basic muscle cell physiology. So we've talked about the general features of the sliding filament mechanism. at the beginning. Then we looked in detail at the neuromuscular junction. Then we looked at excitation contraction coupling, and then we went step by step over the contraction cycle, that cross bridge cycle that physically forms the sliding filament mechanism.